First, we will start by grabbing a large graduated pitcher and filling it with DI water. Depending on how many liters you are making, you will fill it up to about 100 milliliters less than your total volume. In this video, we are making 2 liters, so we fill it slightly under the 200 milliliter mark. This is important because it leaves space to add liquids throughout the process, as you will see later. Next, we will be grabbing our media with sucrose powder included from the fridge. And we will weigh it using a large weigh boat and a scale. The recipe calls for 34.3 grams per liter, so for 2 liters we will be measuring out 68.6 grams. Now we will be adding the media powder to our DI water, and we can use a DI water squeeze bottle to rinse off any extra powder that remains on the weigh boat. Add a magnetic stir bar to the solution and place it on a stir plate to incorporate the solution. Next, we will be adjusting the pH, so you want to turn on your pH meter and grab a container. Remove the pH probe from the storage solution and make sure to rinse off the storage solution with DI water before placing it in the media solution. Place the probe into the solution using a stand. Try to place the probe towards the sides as the height of the water is higher because of the stirring. Once it is in the solution, press read on the pH meter and you can see that it currently reads at about 3.8 pH. Our goal is to get the pH to 5.8, so we need to add a base, in this case NaOH, in order to raise the pH. So first we are starting by adding 1 molar NaOH drop by drop as we increase the pH. Once you get closer to 5.8, you can switch over to 0.1 molar NaOH to increase it by smaller, more precise amounts. Make sure to let it stir for a bit between drops to get an accurate reading of the pH. Now we will be measuring out the agarose, so you want to grab a medium weigh boat and measure out 8 grams per liter of tissue grade agar. We will be adding them individually to each jar, so even if you are making more than 1 liter, you will only measure 8 grams at a time. Carefully add your agar powder into your 2-liter glass jar. Now that the solution is all mixed, carefully remove the pH probe, making sure to rinse it with DI water before returning it to the storage solution and turning it off. Next, measure out 1,000 milliliters or 1 liter of solution. If you are making 1 liter, simply add DI water to reach a total volume of 1 liter, since we started off with less than 1 liter. If you are making more than 1 liter, if we were to use these liters directly after correcting the last one of the liters would be more diluted because it has more water compared to the others. To fix this problem, after we adjust the total volume by adding DI water, we must combine them in another container before measuring out the final 1 liter of solution that will be added to the agarose to ensure proper concentration of media. Once you are done measuring, you can carefully pour the media into your 2-liter jar. Note that even though we are making 1 liter, we use 2-liter jars because in the autoclave it gets hot and boils, so we want to allow space for it to boil and not overflow. Feel free to swirl the jar to stir it a little bit, but the boiling process in the autoclave will combine it. It is important that you crack the lids to allow for gas exchange as it boils in the autoclave. Pick a point on the lid, usually use the words, and turn it 360 degrees. Next, you will cover it with aluminum foil and place an autoclave indicator tape on top. Repeat this with the remaining liters and bring it over to the autoclave once you are done. After you have signed up for the autoclave and it is your turn, place a metal tray and carefully add your jars of media. Feel free to use heat-safe gloves if you feel more comfortable. We will be using cycle 1 because it is the longest to ensure for proper sterilization. To start the autoclave, close the door all the way and press cycle 1, press start, and then log the time that it has been started along with your name so you know when to take it out. After it has cooled a little bit, we will be labeling it with the date it was made. Make sure to fold over a little bit of the tape to make it easier to remove later. The following day, once it is all cooled, you can put it away in the cabinet, but make sure you place it in the back and move the older media to the front so it can be used first. For pouring media, check that there is enough DI water in the water bath and then turn it on. You want to turn it on a little bit before you start melting the media so it can reach 56 degrees Celsius. Remove the aluminum foil and place your jar into the microwave top end first. Then carefully unscrew the lid as far as you can go. Use power level 7 for 11 minutes and then set a timer for 15 minutes to allow for it to cool in the microwave a little before we transfer it to the water bath where it can cool down slowly without solidifying. Next, we will be preparing the laminar flow hood with 70% ethanol. Make sure to spray everything and then turn on the UV light and let it dry for a few minutes to ensure it is disinfected. After the media is done cooling in the microwave, carefully close the lid. It is very hot at this point, so you want to use protective silicone mitts and make sure you face the top away from you to ensure no hot steam or media comes towards you. Place it into the water bath and cover it with aluminum foil to reduce water evaporation. While the media is in the water bath, we will be preparing and labeling our petri dishes. Spray your hands and arms with 70% ethanol. Carefully remove the petri dishes from their packaging, ensuring you are holding the lid closed so nothing gets into the sterile containers. We will be using about 30 to 32 petri dishes per liter of media. Place sticker labels that correspond to the recipe of media we are making. This includes any hormones and antibiotics that will be added later. Make sure to place it on the bottom part of the petri dish, not the lid, and repeat this until you have labeled all of your petri dishes. After about 35 minutes minimum, it should be warm to the point you can hold your hand to it and you can remove it from the hot water bath. Make sure you replace the lid and turn it off. The additives for the media are found in the chemical storage fridge and freezer. Some of them, like LA and AS, are pre-aliquoted in tubes and boxes in the freezer. Make sure you spray everything that goes into the hood. This includes the additives, the media jar, pipette tip boxes, and pipettes. Also, spray your hands and arms with ethanol before entering the hood. Now we will be following the recipe for the media. First, we are starting with 3 BAP, which means 3 times of a 1000 microliter pipette. Carefully add the solution without touching the sides of the jar with the tip, and since you are adding a colder liquid to a hotter liquid, you want to make sure you are swirling to stir to make sure that it doesn't start to solidify before you are ready to pour it. Next, we will be adding our IBA. This will be 200 microliters, so we switch pipettes, and again, you want to carefully add it without touching the sides of any of the containers. Next, we will be adding in the AS. Since these tubes were frozen, hold it in your hand and allow for it to thaw. After it is thawed, add the whole tube since they are pre-aliquoted to 1 milliliter. Swirl and combine, and then repeat this for the LA. Finally, we are ready to pour. We will be using 25 milliliter pipettes, but since we will be filling our petri dishes with 30 milliliters, we will be going to the -5 mark. Carefully put your pipette into the jar without touching the opening of the container. If you do, change to a clean pipette to ensure minimal contamination. Once we have 30 milliliters, place the petri dish and hinge to open the lid. Carefully take out the pipette and pour out the media. If any bubbles are created, place the tip of the pipette on the bubble and the capillary action will naturally pull it up. Once you are done, carefully close the lid and add another plate on top. This is my preference, but this technique varies by personal choice. Repeat the steps for the rest of the petri dishes. If you get any media on the outside of the dishes or on the table, make sure you clean it plentifully with ethanol because since this is media, it is pure nutrients and anything will start to grow and cause contamination. Normally, we do stacks of 5 petri dishes and then carefully push them off to the side to cool. The next day, if they are solidified, label a bag with the date they were poured, the recipe/type of media, and the initials of who made it. Now you want to spray your hands with ethanol and check to see if there is condensation on the top. If so, you want to leave them to dry for another day. If they don't, they are good to be put away. Make sure you tightly hold the lid and bottom closed so nothing gets in while you are transferring them to the Ziploc bags, and then you can put them away in the media cabinet.